Hey there, biology enthusiasts! Today I'm taking you on a journey inside one of the cell's most fascinating factories, the Golgi Complex. First discovered by Italian biologist Camillo Golgi in 1898, the Golgi Complex was initially controversial but is now recognized as a cellular powerhouse thanks to modern imaging techniques. Picture this, a series of flattened, membrane-bound sacs called cisternae, stacked like a neat pile of pancakes right next to the cell's nucleus. Each cisterna is about 0.5 to 1.0 micrometers in diameter, making them teeny tiny but super important. Depending on the cell type, you might find just a few of these stacks or thousands. In mammalian cells, they often form a ribbon-like structure, adding to their complexity. So what does the Golgi complex actually do? Think of it as an organized assembly line for modifying and sorting proteins and lipids. Proteins and lipids enter the Golgi at the Cis-Golgi network, or CGN, from the endoplasmic reticulum. They then move through the medial cisternae, where most of the biochemical modifications happen, and finally reach the trans-Golgi network, or TGN, where they're sorted and dispatched to their final destinations. The CGN acts as a quality control hub, making sure only the right molecules proceed down the line. The medial cisternae are where the magic happens, with enzymes modifying carbohydrates attached to proteins. At the TGN, the finished products are packed into vesicles and sent off to various parts of the cell, but there's more. The Golgi complex is supported by a cytoskeletal scaffold of proteins like spectrin and chirin and actin, giving it structural integrity. Motor proteins help guide vesicles in and out, while a separate fibrous network called the Golgi matrix assists with assembly and disassembly during cell division. One of the Golgi's key roles is protein glycosylation. Enzymes in the Golgi systematically add and remove sugar residues from proteins, creating diverse glycan structures crucial for protein function and stability. O-linked glycosylation, attaching sugars to serine or threonine residues, happens exclusively within the Golgi. The Golgi also synthesizes complex polysaccharides like glycosaminoglycans for proteoglycans and components of plant cell walls such as pectins and hemicellulose. This makes the Golgi central to cellular metabolism and trafficking. Now, how do materials move through the Golgi? Two main models have been proposed: the vesicular transport model and the cisternal maturation model. The vesicular transport model suggests that cargo moves forward from one cisterna to the next via vesicles. However, recent evidence supports the cisternal maturation model, where the cisternae themselves progress from CIS to trans as they mature. In this model, vesicles transport Golgi-resident enzymes backward, not forward. This keeps the enzymatic functions in place as the cisternae mature. While some researchers still support vesicular transport under certain conditions, the prevailing view is that cargo progresses through maturing cisternae, with retrograde vesicle transport maintaining compartmental specialization. In summary, the Golgi complex is a dynamic, multifunctional processing center essential for post-translational modification, sorting, and dispatch of macromolecules. Its organization, enzyme localization, and transport mechanisms ensure efficient intracellular trafficking and are vital for cellular homeostasis. So next time you think about the inner workings of a cell, remember the unsung hero, the Golgi complex. Thanks for watching and stay curious.